Hello, this is a thesis defense presentation that I gave in 2019, about a year ago, and I'm going to do my best to walk through it another time. And I'm going to talk about microbial alteration of volcanic glass. Um, so to do that, I'm going to talk about something called putative biogenic alteration, which is the technical term for the microbial alteration of this volcanic glass. To understand where it's happening, we'll do a little bit of volcanology. I'm going to introduce you to a volcano called Upsal Hogback in Nevada. We'll talk about the PBA in that volcano's tuff. We'll talk about what tuff is first. Then we'll discuss secondary alteration of volcanic tuff and glass. This is basaltic tuff, and the physiochemical constraints that we can elucidate from that secondary alteration, and then the implications of this study. So what is putative biogenic alteration? Well, putative biogenic alteration is a technical term for these textures that you can see in the lower right, and putative means reportedly. We say putative because unless we're observing the microbes, bacteria most likely, causing the alteration firsthand, we have to say reportedly because we are suggesting that that's what caused them. Biogenic is self-explanatory. We're suggesting that these are emplaced by microbes, specifically bacteria, less likely archaea, and alteration are these textures you're seeing in the lower right, these little microtunnels that are emanating from the pockets in volcanic glass. These pockets in volcanic glass are caused by gas bubbles trapped in the magma as it cooled. It was rapidly quenched through interaction with water, which we'll talk about. And when that happens, the gas pockets get trapped. They leave something called a vesicle. Here's a vesicle filled with calcite, the secondary alteration. So what are these textures? They're ichnofossils, which means that they're trace fossils in glass. And we're used to seeing trace fossils in things like sediment, like sands and clays. But in this instance, unique instance, they're occurring in a solid material. And they're caused by eudendoliths. So an endolith is an organism that lives in a rock. A eudendolith is an organism that lives in a rock, in a home, a cavity in that rock, which it made. We'll talk about how they make that. And these are microscopic. The scale of the picture in the lower right that we're looking at, the scale bar says 20 micrometers, so you can see that the textures that are emanating from the vesicles are probably about 5 to 10 micrometers long. They're quite small. And they are attributed to bacteria. That has been studied. You may think perhaps archaea, but bacteria was a better match for what DNA has been found in association with these. And the organisms that cause them are chemotrophic, meaning that they don't need oxygen to perform their respiratory and metabolic functions. They can use other oxygen donors, such as metals. And they're thermophilic, which means that they like to be warm, 60 to 80 degrees Celsius typically for thermophiles, over 80 degrees Celsius for hypothermophiles. So this is the type of thing that you would find in hot springs at Yellowstone. So how do they do what they're doing? The microorganisms, the bacteria, can create a microenvironment around themselves that could be highly acidic or alkaline depending on the organism, and that causes a dissolution of the volcanic glass. So we established that these textures are biogenic or biogenically emplaced based on a few factors. Their size and size distribution, so they have to be the appropriate size to have been caused by the organisms that we suspect. So a couple micrometers in diameter, a few micrometers in diameter. The morphology should be fairly complex. We look for terminal enlargements. We look for bifurcation of the microtunnels. The spatial distribution should suggest colonization. Syngenicity means that they were emplaced at the appropriate time in the rock history, which is reconcilable with a period when conditions would have supported the existence of the appropriate type of life. And also, we're always going to hope to find organic molecules, namely carbon, nitrogen, sulfur, and phosphorus, or ideally DNA, which you can do through a process called DAPI staining. We're not going to get into that in this study. So why do we care about bacteria colonizing this rock type? Well, bacteria were the first life on Earth, and they have populated the Earth for at least 3.5 billion years, long before eukaryotes. And we have evidence of that in the fossil record of a similar type to what we're discussing here. The tree of life is the diagram in the upper right, and at the base of that is something called LUCA. LUCA is an acronym for the last universal common ancestor. And toward that end of the tree of life, what we find are thermophiles and chemotrophs. So those organisms that like to be warm or persist in a warm environment, a very warm environment, and that don't necessarily need oxygen to survive. That is relevant to Earth history because at first Earth did not have an oxygenated atmosphere. That was something that happened much later, largely because of microbes, cyanobacteria, oxygenating the atmosphere. And on early Earth, with an unstable environment, an unstable atmosphere, places like black smokers provided stability, so a place where things were always warm. So of course you're going to have the evolution then of microbes that persist in those very warm environments. Now, why is that relevant to that big orangey reddish brown ball in the left? That's Mars. We can see the scar of Valles Marineris across near the equator of Mars, and we are currently, we being humans, sending machines to Mars to try and find signs of life. And this study is pertinent to that because we may be able to find an analog on Earth that will guide our study or our selection of samples on Mars where we could look for life. Now bear in mind that since these microbes were the first forms of life on Earth, we can presume that they were likely the first forms of life anywhere else. You're not going to spontaneously form an elephant or a dinosaur. You have to get there through the building blocks of life and through evolution from a microbial standpoint. So if life never evolved to the macro extent that we find on Earth with humans walking around, most likely it at least had to go through microbial stages first. So it is the most likely type of evidence of life we would find in the solar system off of Earth. So then the purpose of this study is to find an instance of a lacustrine environment where putative biogenic alteration has occurred and then constrain environmental parameters. This says a second instance because the predecessor in our lab at Brock University, my predecessor, Matt Nikitczuk, working under Marie Schmidt, had found this exact kind of thing in a volcanic center from Oregon, and that was the first continental setting, non-marine. So we sought out to find a phreatomagmatic volcano, which is to say a volcano that erupted in direct interaction with water, standing water, in a continental setting. The methods to do that, we went out and did field observation after selecting some sites to look at. We got our samples and did petrographic observation of those samples, trying to see if we had PBA. Having done that, we did X-ray diffraction, both bulk powdered and micro being in situ, where we're going to shoot X-rays at a thin section, a polished thin section that's about a 30 micron thick slice of rock, and we can elucidate what minerals are in the rock from the diffraction of those X-rays, X-ray fluorescence, which is how we get bulk chemistry of the magma from basalt samples. And then we're going to do some Raman spectroscopy, which is shooting lasers, which is great. And we did that at the Royal Ontario Museum, and some stable isotope analysis that was actually performed at Western University. I should mention also that the micro X-ray diffraction was done at Western, bulk powdered was all done at Brock, as well as petrographic observation. So I'm talking about these volcanoes, and most people don't know a lot about these oddball types of volcanoes. They think about something like Mount Fiji, but all over Earth there are Maars and Tuff rings, and these are types of volcanoes that form from the interaction of magma with water, but in two different environments, and that's what differentiates them largely. To see them, what differentiates them is their morphology, and the names reflect that. So the Maar is basically a crater in the ground, and that crater forms because as magma rises through a conduit, it interacts with groundwater. And when that happens, there's a rapid expansion of the water as it is cooling the magma and absorbing the heat, and it gets converted to steam. Of course, it wants to expand, but it's constrained by the ground, and eventually that's going to lead to an explosion. And that explosion is going to it's going to excavate all of this material, and there's going to be lots of bits of country rock in the deposits surrounding the Maar, and it's going to have somewhat of a ring around this deep crater, and it's going to have a lot of what we call accidental clasts, which is the country rock. Tuff rings begin in a very, very similar fashion, except once the magma has reached the surface, whether it's subaerial or understanding water, well, it would be understanding water in this instance. It's going to continue, the magma is going to continue to interact with that standing water, and it's still explosive, but it's not constrained by being subterranean anymore. So what you're going to get is explosions of steam and ash, and we're getting ash because it's a very efficient eruption, meaning that as that magma is quenched, it's going to freeze into primarily glass with some rock. Now, glass is what we get when we don't let crystals nucleate as a rock cools. So it's amorphous, it doesn't have a long-term crystal structure, and efficiency refers to how fine the particles are that are produced by the eruption. And when they get rapidly quenched like this, super cooled, you're going to get a very efficient eruption producing a lot of ash particles that are very small. So then what is tuff? I'm going to say tuff a lot because that's the type of rock that we're looking in. And tuff is referring to largely the size of the clasts of our material. And in a rock that we call tuff, we have over 75% ash, which means very fine particles at the lower right of this pseudo-ternary diagram, and lesser components of lapilli, which are clasts that are 2 to 64 millimeters in size. Those are these smaller black chunks that you can see in this picture of tuff at the upper right. And of course, we're going to get some blocks and bombs, which are larger fragments. So anything 64 millimeters and larger are blocks and bombs, and that's where we get our geochemistry from. That's what we grind up and have X-ray fluorescence performed on. So when I say tuff, I'm talking about what's in this picture. It's mostly ash-sized particles, less than 2 millimeters, and it's got some lapilli in it. Specifically, we're looking for a vitric tuff. But pretty well most tuff you're going to find is vitric because it's been frozen, solidified very quickly, which means it hasn't nucleated crystals, particularly in basaltic eruptions involving water, so it's mostly glass. So this is very fine glass particles that have then been left to stand and hopefully have become nicely indurated. Indurated meaning that they're solid, not friable. Friable would be something that comes apart very easily. Indurated means it's been cemented and a little bit compressed and it's fairly solid. So a little bit more on what kind of tuff we want. Well, I said we want a vitric tuff, so we want a lot of volcanic glass. Since we're looking at basalts, that volcanic glass is called sideromelane. But more to that, we want palagonite. Now, palagonite is a secondary alteration of volcanic glass that occurs in the presence of warm water. And what happens is the glass becomes dissolved a little bit, incongruently or congruently depending on the chemical conditions, but you're going to first form a product called gel palagonite, which in this top microphotograph is pictured away from the vesicle. And eventually, if we dry it out a bit more, that gel palagonite, which is also amorphous, doesn't have long-term crystal order, is going to start forming fibropalagonite, which if you look at the lower photograph in cross polars, does stay visible, and that's because it's composed of clays, things like smectites. So then we had to select a site, and where are we going to go? We selected something in the Great Basin. The Great Basin is an extensional system in western North America that lies between the Wasatch Range to the east and the Sierra Nevadas to the west. Largely we're in Nevada, and specifically we went to the Lake Lahontan Basin and the Carson Sink in that basin. Now, Lake Lahontan, if you go looking for it, you're not going to find it today. It's desiccated. It was a pluvial lake, which means that it existed at the end of the last glaciation, largely because evaporative conditions were different than they are today, and the accumulated water coming off the mountains didn't evaporate. And that was up to about 10,000 years ago when it started evaporating rather quickly. This was a very large lake. The surface area was about the same as Lake Ontario and it was about 250 meters deep in places. So it offered, with a little bit of volcanism, a nice environment to try and find some volcanic glass. So within that basin, we selected Soda Lake, and Upsal Hogback as potential target sites. Soda Lake did not have well indurated tuff, and it didn't have altered tuff. Upsal Hogback did. So Upsal Hogback became the focus of this study, and that's where most of the work was done after initial field work.